All right, guys, mutations this week. We're going to do mutation repair next week. Keep yourself oriented. We're talking mostly about DNA. That's going to be the main point of looking at mutations. We want to know, do you have any mutations happening in your double-stranded DNA? Because we want our DNA to stay consistent. We don't want it to change. Now, if you had a mistake occur in your messenger RNA, like your RNA polymerase transcribes it wrong and puts the wrong nucleotide in, that's probably not as big a deal because, yes, it's messed up and it's going to make a messed up protein, but you can get rid of that messenger RNA pretty fast. They're going to degrade within a day-ish, if not hours. Whereas your DNA is going to stay constant like forever. So we don't want mistakes happening here. So we have lots of ways to repair that, and that's what we'll talk about next time. So just to keep ourselves oriented, and remember, every 3 nucleotides we call a codon. And so these are our codons. So these are our this would be our coding strand of DNA and our messenger RNA on this side. And so remember, you can start in the middle and move out. So we say like, oh, guanine, guanine, guanine makes glycine, or guanine, cytosine, adenine makes an alanine. Okay, and so these are our amino acids, and usually we're going to think about our mutations in terms of the effect that they have on the proteins. You don't have to think about it that way, but that's the most useful way to actually think about it, okay? So when we have a mutation occur, like if I switch this G, let's say it's a GCC, okay? So if I change this G to an A, now it's ACC, right? So instead of GCC would be alanine, it would be ACC, which is threonine. Those are different amino acids. That's going to change the actual structure of the protein, which we talked about last time. Now, mutations are just the changing of the order of nucleic acids. Now, it could be a good thing, not usually. It could be neutral, sure. That's fine. Yeah, maybe it's neutral, but usually it's harmful because you're messing with those proteins. We don't want to mess up our proteins. Now, it's caused by a variety of things. Radiation is what we usually think about, but there are tons of chemicals that can cause mutations. And actually, one of the main ways that mutations get made is by having your DNA polymerase make a mistake. But we have really good ways to fix that, so that's really nice. So let's get into it. So usually what we're talking about are point mutations. These are also called substitutions. I always called them point mutations. But you're just going to change one. We're going to focus on changing one nucleotide, okay? So we're either going to like completely change what it is, so are we switching from a purine to a pyrimidine, or can we just kind of alter it a little bit to change how it's bonding, okay? And that's just going to depend on the actual mutagen itself. Now, usually we categorize that, like I said, based on the protein's function. You can categorize it based on what you actually changed it to. So if I change it from a T to an A or a G to a C, those are called transitions and transversions. I'm going to give you a little chart that talks about that. But usually we talk about what it's actually doing to the amino acids that are going to result from the messenger RNA. Now on this chart here, what I actually have, so up here, this is the non-coding strand, so this is the template strand, okay? And then we're going to make our messenger RNA off of that. So if you had a thymine, it would match up to an adenine. If you have thymine, adenine. Cytosine, it matches up to a guanine, okay? So that's important to remember. This is the coding strand. That's why these would be the same. So, but we make it off of the non-coding strand or the template strand, right? So it's complementary. Anyway, so let's focus on the messenger RNA here. So I've got our adenine, our guanine, that forms a lysine. Now, all we really need to know, if you've taken biochemistry or if you're taking it right now, you know that there are a bunch of different amino acids. There's 20 that you need to know. They have different sizes, different charges, and all that stuff. If you have not taken biochemistry, that is okay. All you need to know is that there are a variety of amino acids. They're all different. They all have different R groups that stick off, and they all have different jobs, really, if you want to think about it that way. Some are non-polar, some are polar, some are big, some are small. It doesn't really matter. All that really matters is that some of them have charges and some don't, okay? And that's where we're going to learn about conservative and non-conservative missense mutations. So, but that's all you really need to know. You don't really need to like have all of them memorized for this class. Now, if you're going to do a point mutation, so let's say we've got our adenine, guanine. Okay, awesome. So if we change the DNA up here to thymine, thymine, so that means the complementary sequence would be AAA, okay? That still codes for a lysine. So let's back up here. So if I change it, so if I have A, okay, 1, 2, 3, that forms a lysine. So if I mutate it from AAG, it's still a lysine. So I'm still making the same amino acid. There's a redundancy built into your actual amino acid codons, which is really, really nice. And so when we have that happen, we call those silent point mutations, okay? Now, We'll get to synonymous and non-synonymous in a second. But that would be a silent mutation. Okay, the actual amino acid in the amino acid chain or your polypeptide chain has not changed. Okay, that's silent. Now, if we actually start changing what happens, there's really two categories of that. There are nonsense mutations and there are missense mutations. Nonsense mutations are what happened when we accidentally make a stop codon. So if you look here, there are three stop codons. Okay, there's UGA, UAA, and UAG. Okay, so if I actually switched, so let's say I had UAC, it was making a tyrosine, and I mutated this cytosine into a guanine, so I'd go UAG, I have accidentally made a stop codon. So now we call that truncation. I have taken a protein that's supposed to be, let's say 100 amino acids long, and I accidentally make a stop codon halfway through. Well, now it's halfway as big, so it's been truncated. We call those nonsense mutations because the protein that actually gets made doesn't make any sense at all. It's nonsense. It's half of what it was supposed to be, or a quarter of what it's supposed to be. It just kind of depends on where it occurs in the actual sequence. Now, the other change that can happen, which I don't know, makes a little more sense is, not that this doesn't make sense, is a missense mutation, okay? So instead of nonsense, which means I accidentally make a stop codon, a missense is just where I change the resulting amino acid, okay? Now, so in this context, that would be like, instead of making a stop codon, like if I had UAU and I made a tyrosine, if I switch that, like the second nucleotide, so if I change it to UCU, it makes a serine, okay? So it could be really bad. It might not be that bad, okay? We haven't truncated the protein, though, so that's good. So our protein will still be the same length of amino acids. Okay, awesome. Now, the problem is, what if I switch from, let's say I was making a really charged amino acid. So lysine is a charged amino acid. So you can see we've got this amino group here with a nice positive charge, okay? So it's charged. I could switch it to another positively charged amino acid, like arginine. Okay, awesome. Now, you can see size-wise, they are different sizes, okay? So lysine is much smaller than arginine, but they do have the same charge. And when you're trying to fold your protein up, yeah, the size might not work super well, but you might be using those charges to kind of hold it together. Maybe this positive charge is going to match with a negative charge somewhere else on the protein. And so you can still have similar folding patterns, okay? So you might not lose that secondary or tertiary structure. Okay, awesome. But, so we have conserved our charge, okay? So that's where we have a conservative missense mutation. That means that I have conserved the functionality or the abilities of this amino acid. So if it's charged, usually we're just saying, is it charged or not, and what charge is it? So if it's positively charged and I maintain that positive charge, okay, it's a conservative missense. If I change what it is, so if I accidentally switch it to like a threonine, which is an uncharged, a non-polar amino acid, well, I mean, this one is polar, but it's not charged, okay? Because you still have that. You would be able to form like a hydrogen bond here with that alcohol group. So it is polar, but it is not fully charged, so that's pretty different, okay? So if I'm not staying within the same charge realm, then it's non-conservative. So we would call that a non-conservative missense. The conservative referring to the actual ability of this amino acid. Now, if I make a protein and I change it in some way, okay, so I have not had a silent mutation, but I have changed the protein, we call those non-synonymous mutations. So a nonsense point mutation would definitely be non-synonymous. A missense mutation, be it conservative or non-conservative, would also be non-synonymous, okay? Now, hypothetically, you could have a conservative missense mutation that doesn't really alter the protein's functionality at all. In that case, I suppose you could call that synonymous, but I would argue that it's probably going to have some kind of detrimental effect, so we would call that non-synonymous. So synonymous and non-synonymous are referring to the actual like abilities and functionality of the resulting protein, okay? I don't like these terms that much. They're not typically used. Usually we're referring more to like what's actually happening in that DNA. Is it a point mutation? Is it silent? Is it nonsense? Is it missense? If it is a missense, is it conservative? Is it non-conservative? So those are the biggies that you need to know. And I even added like a little extra point here that yes, you could have a missense that doesn't really alter the protein's functionality, and thus it is not non-synonymous, but I don't know. And most of the time it's going to be. to give you just another visualization of what's actually happening. So up top here is our normal sequence. So we've got our RNA with our anticodon. So this will be your tRNA that matches up to the actual amino acid. Okay, so I like this chart a lot. It's kind of nice. So you can see when we have our missense mutation, down here we're going to change this guanine right here to a cytosine. Okay, so that is a missense mutation because we haven't accidentally made a stop codon. Awesome. And then that's going to just change your protein. Now, if it's like non-conservative missense, man, you can really mess up with that protein. So here, like they're just trying to emphasize that if you have this protein that's folding like this, and I change that amino acid, it can really mess up the protein structure. Now I have a nonsense down here. So it was supposed to be AUA, coding for what is that, isoleucine. And I changed it to UAA, and it really messed things up. It accidentally made a stop codon. And now my protein is like half the size it was supposed to be, and that's called a nonsense mutation. Now, I mentioned usually we categorize mutations based on the effect that they have on proteins, this is obviously where those names come from. Like missense is referring to the protein, nonsense is referring to the protein. You can also refer to mutations, point mutations, based on whether they are transition or transversion, okay? And usually we would say like it's an A to G transition or a C to T transition. So if you're going between purines, okay, because they're not that different structurally. I mean, they have some big differences. You got that nitrogen over here with this carbonyl, and then you got nitrogen here, nothing over here. So they are pretty different. But if you're transitioning between purines, that would be slightly easier than transitioning between like a purine to a pyrimidine or vice versa, because you have all these extra carbons, you have a whole extra ring. So are transversions less common? Probably. Yeah, so just this idea, if you're transitioning between a purine, you're transitioning between a pyrimidine, but a transversion is going between a purine to a pyrimidine. Okay. Honestly, unless you're dealing in this kind of mutation study of these types of point mutations, usually we're more concerned with looking at the actual resulting protein. Now, yes, you can change your nucleotides, okay? So that's totally fine. That's a point mutation or a substitution. But what you can also do is you can add them where they're not supposed to be. And we call those additions or deletions. And of either one or two nucleotides. Now, there could be more than that, but I'll get to why we don't typically refer to those as frameshifts. So this idea is called a frameshift. So your frame is the term of use for the actual order of your codons, okay? So every 3 codons makes an amino acid, right? So that's my frame. So if I have 3 U's, I make a phenylalanine. If I have a UAU or UAC, I make a tyrosine, and so on, okay? So that is my frame. If I stick a nucleotide somewhere in here, it's going to shift all of these nucleotides over, okay? And so in this case, here's a substitution, but let's say right here. So in this case, I have an insertion. So I'm going to, there was a C, I'm going to stick a C in here, okay? And it's going to shove all my DNA this way, and that's going to change all the frames of the subsequent codons, okay? So instead of, accidentally just changing one amino acid, now I have changed all subsequent amino acids. Okay, so that's just if I add one nucleotide where there wasn't one before. Okay, so that would really mess things up. Now, you could also delete them, and so those are called deletions. Now, a frame shift has to be either 1 or 2, because if let's say I added 3 nucleotides, or I deleted 3 nucleotides, well, that would only change like 1 amino acid, okay? So I would either completely delete an amino acid because that whole codon is gone, or I have added a complete amino acid because I added 1 whole codon's worth. We don't actually call those frame shifts. So if there are 3 nucleotides, that does not count as a frame shift. But if there were 4, yeah, sure. But usually we just say, you just refer to it as like, did I add 1 nucleotide or did I take 1 nucleotide away? So is it an addition or a deletion? Okay, so that's what a frame shift is. Now I have a little video that I wanted to show. It's very generic, but just to kind of visualize what's going on.
DNA rarely makes mistakes, but it can happen. A point mutation changes a single base. This can change the whole amino acid, or depending on its position, could go unnoticed. Other mutations can be more damaging. A deletion removes a base, but also causes a frameshift, which changes every amino acid after the mutation. Originally, the codons would be formed in this pattern. But after the deletion, the amino acid sequence will change because the bases shift by 1. The resulting protein will likely be non-functional. An insertion adds a base, but also causes a frameshift, which changes every amino acid after the mutation. Originally, the codons would be formed in this pattern. But after the insertion, the amino acid sequence will change because the bases shift by 1. Again, the resulting protein will likely be non-functional.
A YouTube channel called Teacher's Pet. So check her out. I think she does other videos too. So one last thing before we actually get into the causes are the idea of a single nucleotide polymorphism. Okay, that is a mouthful, I know. We call them SNPs. Okay, so that's what we typically would refer to them. Like if you were, being some nerd somewhere at a nerd conference, you'd call it a SNP. So a single nucleotide polymorphism. So usually when we're thinking about mutations and all this kind of stuff happening, that's like you think about one of your cells got exposed to an X-ray and it caused a mutation in one of your cell's DNA. Okay, fine. If you have a mutation that occurs throughout your body, okay, and it's in a large substantial portion of the population, so it's not just you. Let's say usually we say it's at least 1% of the population. Okay, then we would call that a single nucleotide polymorphism. So we would say, all right, I like this chart a lot. So let's say you have a cytosine and a guanine matched up in your DNA. Okay, 95% of the population has that. If we have a SNP in 5% of the people, maybe that's different. So now it's an adenine and a thymine. Okay, so that's what a single nucleotide. Okay, so it's a one nucleotide location, right? So a single nucleotide. Poly, meaning multiple, morphism, so it's like different. So at one location, we have a different nucleotide. So that's a single nucleotide polymorphism. So it's kind of like a mutation that wasn't detrimental, that just kept getting passed on to subsequent generations, but it's not the most common one in most of the population. So it could be like a mutation that causes some kind of disease, but you can still generate offspring. It's not that big a deal, or like let's say this would cause, this is not one of them, but like 5% of people have red hair and the rest of the population doesn't. So that would be a single nucleotide polymorphism, because having red hair wasn't necessarily a detrimental mutation. It just, was carried on. And so we call that a SNP. Lots of people have SNPs. There's SNPs everywhere. Usually the ones that we're referring to are synonymous. So remember I talked about that. Synonymous being silent point mutations. Synonymous, as you recall, refers to, like, does the protein still do its job? It doesn't have to be a silent mutation. It could be a missense mutation, as long as the protein is fine. And so, are there any problems? No. Okay, then it's synonymous. But you can have non-synonymous SNPs, and that's how you can get mutations in people. Like maybe you have a taste receptor that doesn't work, or you have some kind of enzyme that's not doing its job properly. Yeah, so it could depend. Maybe you're truncating an amino acid improperly, or a polypeptide chain improperly. One of these that makes me think of would be the truncation of the CCR5 co-receptor for HIV. So a bunch of your cells have CCR5 on them, your T helper cells. And some people, a small portion of, I think, Europeans can have a truncation. So we call them, it's a deletion mutant, but their CCR5 is truncated. It's not the correct length. And they're actually resistant to some strains of HIV because of that, because HIV needs you to have a full length CCR5. And so, but that's a really small portion of the population, probably because it hasn't really been selected against, or selected for in this case, because not everybody gets exposed to HIV, so why would you select against that mutation? And so, but it does exist in the population, so we would call that a SNP. Now, it could have more than one nucleotide having to do with it, but it could just have one. I honestly don't know off the top of my head, but that would be generating a non-synonymous nonsense mutation. Now, the fun stuff. We need to talk about, not that this isn't fun, we need to talk about how we actually get mutations. Now, there are a few different ways, and I'm going to try and take you through all of them, okay? So, one of the biggies, we're going to start here. This would be your own body is causing these, okay? So, this would be replication error. So, polymerases, be they DNA polymerases, be they RNA polymerases, they make mistakes, okay? They can put the wrong nucleotide in order. So when they build from 5' to 3', right, because they always build 5' to 3', doesn't matter the kind of polymerase, they can make mistakes. They put the wrong nucleotide in there. Now, RNA polymerases can make mistakes. I've said this before. It can be detrimental to the actual messenger RNA and the subsequent protein that it's making, but it's not going to like hurt the actual creature itself because the messenger RNA is going to degrade eventually anyway. DNA polymerases, yeah, that's not so good because you're messing up your DNA. Now, a term of use is fidelity. So if you're doing like a polymerase chain reaction, that's called PCR. That's one of the ways that like we would amplify DNA for coronavirus to see if there's a coronavirus genome inside of your saliva or something. You would use PCR to do that. The actual polymerase that we use for that process is something that we isolated from a sea creature. It's a bacteria in the sea, and we pulled their DNA polymerase out. And it has fact-checking ability, okay? So how accurately can it actually replicate a strand? So that's your fidelity. Most DNA polymerases have pretty high fidelity. If you're doing like scientific work and like working in a lab and you're doing PCR and you need to make sure that when you do PCR, it is exactly the same strand every single time, they sell high fidelity DNA polymerases that have super awesome fact-checking ability and they hardly ever make mistakes. Your DNA polymerases tend to have pretty high fidelity. Other creatures, not so much, especially in viruses. So like HIV, for example, because that's what I know, it can use its DNA-dependent RNA polymerase activity from its reverse transcriptase. It has poor fidelity. It has like zero. Well, I shouldn't say zero. It has really, really poor fact checking. It won't check itself. So it has poor fidelity, which makes mutations happen a lot more often, which for a virus is a good thing because they want mutations to occur because that lets them evolve more quickly. But you don't want that to happen because it turns out we don't make a lot of babies, and so we can't evolve very quickly when we have mutations. Usually you just die. So when we add an incorrect base, what can happen is you actually shift the shape of your DNA polymerase. Okay, and what that does is you have kind of two domains of the protein. If you haven't taken biochemistry, that's okay. You just have different domains. So you have your DNA polymerase, and you're doing your polymerase activity. You're adding nucleotides to that strand, and you accidentally put the wrong one in. Well, that changes the shape of it and shifts the whole strand into an exonuclease domain. And we're going to talk more about that next time. That's how we actually fix these problems, okay? But that's not what we're talking about today. But I did want to just show you the actual error rates that get made, how many errors occur when you have exonuclease activity. So that would be your fact checking or your base repair ability in a DNA polymerase versus if you don't. And so this was a chart here from a paper. I think these are both from the same paper, where they were calculating the error rate, like how often does a DNA polymerase actually make mistakes. And you've got humans here versus yeast. SC is for yeast, HS, or Homo sapien, Saccharomyces for yeast. And you can see down here we have exonuclease proficient. So this is how often do we have a substitution? Okay, so we talked about that. So this would be a substitution where I actually change the nucleic acid versus a frameshift, like an addition or a deletion. So that's what single base indels, that's just a term for an insertion or a deletion. And so you can see how often does that happen? Well, error rate times 10 to the negative fifth. So it doesn't actually happen a ton, but remember, your DNA strand is crazy huge. There's like, what, how many? Millions and millions and millions of nucleotides. So when you start, 10 to the negative fifth, that's actually a lot when you start thinking about how big your actual genome is. And so if you have fact-checking ability down here, look at this. So how often do you get an insertion? Not very often at all, okay? 0.05 times 10 to the negative fifth. And we have other repair mechanisms that we'll talk about. So that's what makes up for the fact that, 1.3 times 10 to the negative fifth times, it actually does happen. But if you don't have that fact-checking ability, look how often this happens, okay? So we jump up to 5, You can have in this the epsilon polymerase, you can have 24 times 10 to the negative fifth base substitutions. That's super high versus down here, if you have the exonuclease present, it becomes practically zero. And so that's why it's really, really important. So your DNA polymerase is really, really cool. And that fact checking ability is really important. Now, it does have mistakes that happen. Okay, so hence why these are not, below zero, or these aren't zero. And we're going to talk about that next time. Okay, let's keep going. So radiation is another big one. So we're moving from inside the cell to outside the cell. So if you suntan a lot or you go to the beach a little too much, you can get lots of different UV light that will hit your skin. It hits your DNA, and it can cause bonds to form. Okay, so there are different kinds of ultraviolet light. They can all cause damage, but UVA and UVB tend to be the big ones that we talk about. I believe these stop signs are referring to, I think, suntan lotion, but I'm not 100% sure. The paper wasn't super clear on what it meant, but it doesn't really matter. The whole point is that you can have this UV light come down, and what it can do is it can form bonds between nucleotides. So this would be a nucleotide, 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 okay? They're not supposed to be bonds here, okay? So you can have either like one bond form here, or you can have a couple, so they're really stuck together or not as stuck together. So this would be cyclobutene pyrimidine dimers, right? Okay, versus this would be a 6,4 dimer photoproduct, but basically between your pyrimidines. So your pyrimidines are your cytosines and your thymines. Now, a lot of times we'll refer to what kind of damage does UV light cause? Thymine dimers. Well, that's not 100% true. You can have, absolutely, you can have two thymines stuck together. You can have two cytosines stuck together. You can have a cytosine and a thymine stuck together. It doesn't really matter. As long as they're pyrimidines, they can form these bonds in between themselves. And what that does is, as your DNA polymerase is chugging along, or your RNA polymerase, but as they're chugging along and they're looking at this for they're going to make a complementary strand, either a DNA or RNA, then they will count this as like one base. So it's sort of like a deletion. And so that would cause a frame shift in the resulting transcript, and thus in the resulting amino acid chain. And that could be really, really bad. Now, there are other types of radiation. So that was ultraviolet light. The other big one that we talk about would be X-rays and gamma rays. And so they don't have particles, but they have a ton of energy. And so I apologize for the quality of this, but this is honestly the best one that I could find. So you have that energy coming in. It's going to hit one of your valence electrons, and it gives it so much energy that not only does it jump to an outer valence orbital, it actually gets ejected out. Okay, and so what is that going to do? Well, that's going to, you just lost an electron. That's a whole negative charge on your atom. And so you've gone from a neutral charged atom, so in this case it's sodium, to a positive charge because we lost that negative charge from that electron. And so we have accidentally made an ion. Sorry, I had a brain fart. So you've made an ion, a charged atom. That's not good because this sodium is now very, very upset, or whatever it is. So it could be a carbon in the backbone of your DNA. It could be that phosphate. Maybe you threw you made some of the electrons, which the phosphorus in the sugar phosphate backbone of your DNA already has a bunch of electrons there that you can excite and make them jump away. And so now your phosphate is super PO'd because you just you've made it go from negative to neutral, and it doesn't like it. It wants that charge. And so it's going to start reacting with as many electrons nearby as it can to fill that orbital again. And so that's how you can get crazy things like really severe damage, like double-stranded breaks or single-stranded breaks, which on the next slide, I actually have a picture of what that looks like. So here's some chromosomes down here. Let me just make it bigger. Here's some chromosomes. And you can see that hole isn't supposed to be there. So these are single-stranded breaks. That is going to give you a real bad day if you have that happen. And that's what X-rays and gamma rays can cause. Okay. And so down here, I actually have, so in blue are gamma rays. In orange are carbon ion beams. I have a couple charts here talking about this. Usually we think about radiation, especially high energy radiation, as being like bad, like Chernobyl, or a nuclear bomb went off or whatever. It doesn't necessarily have to be bad. So sometimes especially for plants, what we'll do to cause mutations that could hypothetically become beneficial mutations, we will use gamma rays. Classically, you would use gamma rays. Sometimes you use carbon ion beams. So they're just charged carbon ions, and it can pull the electrons away in the exact same way. They have different functions. If you want to read more about it, I have a paper right here that you can read about how they actually do that in plants. So that's what orange is referring to. But look at blue. So here, I just talked about transitions versus transversions, right? And so it turns out if I just start throwing energy at these electrons and making them jump out, I can cause transitions from like adenine or thymine to guanine and cytosine, or vice versa. Or I can cause a transversion. So more commonly you see transitions, okay? That actually makes a lot of sense, right? It's easier to go between a purine to a purine or a pyrimidine to a pyrimidine versus jumping between these. But you can do it, it's just not as common, okay? So let's talk a little bit about the kind of damage they can cause. So when you have different amounts of radiation, the point I wanted to hit here was we're going to talk more about DNA repair later. But when I introduce the amount of energy, so in this case, where the energy is going up as we go to the right, and we're going to talk about how often, I mean, we're talking about it now, incidence would be, so GY is grays, so that's just a measure of radiation. It's like rads. It's just how much radiation is actually getting in there. And why did I put this in here? Well, I just want to talk about, we're going to talk about excision synthesis, we're going to talk about recombination repair, we're going to talk about non-homologous end joining, as ways to fix the actual damage caused by gamma radiation or X radiation. But if you have these single base damages, you fix that super fast within a couple minutes. Single stranded breaks, not so great. It takes a little while to fix that, if you can even fix it at all. Double stranded breaks, way bigger deal. It takes a lot longer to repair that. But the actual amount of damage depends. So, I mean, this chart is actually really obvious. If you throw more radiation at a piece of DNA, it's going to cause more severe damage. Yes, that's like the whole point of having this chart on here. Now, look here. What I wanted to talk about, last thing with high energy radiation. So this is from an Arabidopsis, so that's just a classic scientific plant. So we'll use Arabidopsis to just do plant studies. And so what you'll do is, so in black is survival rate of the plant, and on here is how much radiation it's actually getting in grays. So it's going up. So it turns out, hey, if you irradiate something enough, it dies. I don't really see why that's that exciting, but I mean, hey, somebody had to prove it, right? So their survival rate goes way down as your grays increase. They're just comparing two different kinds of plants. Or no, it's gamma versus carbon. That's what they were doing. They were comparing gamma ray radiation as a way to create mutations versus carbon ion, and just, you know, how different it is. But look here. So I've got my grays, my gamma rays go up, plants die, and an important thing to remember is that your fertility also goes down when you are exposed to radiation. And so this is the actual fertility here in the plant. What's it doing? Well, usually what you're doing is you're destroying the DNA, and you're actually destroying the chromosomes, like the sex chromosomes, or you're messing with its ability to form pollen, or in humans, like you're messing with their ability to form sperm or eggs, though eggs are already formed. So it's usually sperm that we think about. But yeah, so fertility drops as well as survival. I mean, this is another one of those duh kind of things, but hey, we got to talk about it. UV radiation, X-rays and gamma radiation, so that high energy radiation. Now let's move on to chemical causes. So there are tons of these. There's tons of charts, just all kinds of different chemicals that cause different things. So usually they can either themselves look like a different base. We call those analogs, okay? Or you can mess up the actual structure of the nucleic acids themselves, okay? And that can cause a variety of things. So you can cause additions, you can cause deletions, a lot of substitutions. Basically, what you're doing is you'll accidentally modify one of the nucleic acids on one side of the strand. And then when you have a replication occur, it looks like the wrong amino acid. So in this case, you have an adenine, you deaminate it, okay? So you make that amine group go away, and you replace it with what is that, a carbonyl? And then when you actually have replication occur, it looks more like a guanine. And so the new strand that you make, the new complementary strand, would have a cytosine instead. It was supposed to have a thymine, but now it has a cytosine. And so you've gone from an adenine-thymine pair to a cytosine-guanine pair. which would be really bad. You've just caused a substitution mutation. And so you can have deamination is really common. You can have alkylation. Okay, so those are, they'll basically take that carbonyl and stick that carbon here. So see, we've got a double bonded oxygen here, and you're sticking a carbon onto there. So the oxygen is still there, but now we're causing different kinds of bonding to occur or potentially occur, because they're not always the most preferential. It's hence why it says a rare tautomer. But basically, you can accidentally make the wrong nucleic acid bind. Okay, I keep saying amino acid. I mean nucleic acid. Yeah, so lots of chemicals can do that. Bad news. Now, usually that's bad. We think about, like, I don't know, like, I grew up in Michigan near Dow Chemical, and they were accidentally releasing stuff into the environment, and so people were getting cancer, and, lawyers are good. They can give some money to people, so they stop suing them. But there are some good ideas, too. So let's say you have cancer, and you want to kill that cancer. Well, a great way to kill cancer would be to use some kind of mutation or some kind of chemical that causes mutations. And so in this case, we can use cisplatin. There are a bunch of these. So these would be chemicals that you would use for chemotherapy, for cancer chemotherapy. And so cisplatin is one of those. So you can see there's platinum right there. That's kind of weird, right? Platinum with some chlorine, hence why it's a pretty toxic thing to use. So you would only want this to, like, really go near the cancer. What you would likely do is you would do multiple therapies. So you irradiate the cancer in the hopes that you can disable some of its repair mechanisms, and then you could throw in a chemical mechanism to actually cause another mutation. And so you kind of attack it a couple different ways, and then it can't fix the mutation, and it initiates, like, apoptosis, basically, is what you're trying to do. But you can see it's a little, how do I say this? Like, it's unwieldy. It's really hard to control oncology. How do you actually treat a patient? Well, it's kind of unwieldy. Like, you're throwing radiation and these chemicals. There's a lot of bystander effects. So that's why treating cancer patients can be really, really hard. But this idea is really interesting. So when you have cisplatin, it basically makes guanine residues either on the same side of the strand or on opposite sides of the strand. They stay stuck together. So you're, like, bonding between them and holding them together. So you can't, like, have DNA replication occur. So your cell either is going to die because it can't do that, or it at least can't replicate, and thus cannot undergo cell growth. Hence why it would be, because that's what cancer is, right? Uncontrolled cell growth. One of the last things before we get into your cells actually using this inside of them. So free radicals, you may hear about this. There's so much misinformation about free radicals, and, like, oh, you need to, I sell nutritional supplements that get rid of free radicals. It's really good for you. My mom used to be super into that kind of stuff. Like, you gotta eat your super fruits, like blueberries and acai berries or whatever. Okay, we need to talk about free radicals a little bit. So free radicals are when you accidentally have, like, an extra electron sitting there, okay? So what's the radical? Well, see that little dot right there? That electron is not supposed to be there, okay? So it's not balanced out. What you're usually going to have is a free radical will perform an oxidation, okay? So they will oxidize something. And so they'll pull electrons away from stuff, okay? Because oxidation is the loss of electrons, reduction is the gain. And so they will oxidize something and themselves become reduced. And so free radicals are made all the time. We make them all the time. They're byproducts of tons of different processes. Your cells use free radicals constantly. All of your immune cells actually have free radicals contained inside of special vesicles to use them as weapons against bad guys. And so we deal with free radicals constantly. This is my one rant about nutritional supplements and all that silly stuff. So can getting rid of free radicals help you? Absolutely, because free radicals will readily attack your DNA if they can get close enough. Okay, but the problem is, like, consuming a bunch of blueberries, is that really going to reduce the ability of free radical? Like, just think about the scale of how many cells are in your body, how many blueberries you'd have to eat. Like, is it really going to have that effect? No, like, I don't know. I'm getting upset. So, if you can have this just happen, I don't know. This is just how, sometimes free radicals get close to your DNA and cause a mistake. That's why we have repair pathways, right? And so, like, I don't know. It's not usually a big deal. We fix this all the time without having to eat a bunch of blueberries. Now, if you have Radiation itself can cause free radicals to occur. And so that would be like unnatural free radical formation. So that's what you can probably avoid. Don't go visit Chernobyl again. I told you to stop doing that. Don't eat alpha emitters. They'll kill you. So that's not only can radiation directly damage DNA, but it can cause the production of free radicals, which can damage your DNA. Okay, so that's the point of this sentence. Now we're cruising. We are almost done. I don't want you to think that all mutations that are made are always bad. Okay, there are a couple instances where we actually purposely will cause mutations inside our cells. Now, one of those, since I'm an immunologist, I got to talk about this, is called somatic. Okay, so it's not in your sex cells, it's just general somatic hypermutation. Induces mutations in antibody genes. Why am I just reading this to you? Induces mutations in antibody genes to increase binding site diversity. Okay, so when you have antibodies, okay, there's your antibody. Okay, this is the business end. These two little spots will grab stuff. This is the end that can sort of connect to like a macrophage or something and tell it, hey, I bound onto something. You should grab this and suck it up and eat it. Okay, so the business end of antibodies is this end. It's what grabs onto bad guys. So I want to have high variability here. And so what happens is I have a bunch of different genes. Okay, so these are your genes. There's V, D, and J segments, okay, for the heavy chain at least, and then for the light chain, there's a V and J. And what you can do is pick different genes. Okay, so depending on the kind of creature you are, you have different amounts of these, but there's a bunch of these. So you can pick a bunch of those and you rearrange them and you pick different genes, and it makes the structure of this end look different. Okay, different binding abilities, which we talk about in biochemistry. So basically, you're always trying to increase the binding affinity. So you want a really big Ka, your binding affinity, and you want a really low Kd, or dissociation constant. Okay, and so by doing that, well, okay, so you pick a bunch of genes. Awesome. Okay, and you're going to make a protein from that. Okay, great. And there's a lot of variability you can get from that. But to increase the amount of variability, what we can do is you can use a special enzyme called AID. It's activation-induced cytidine deaminase. So what is it going to do? Cytidine, okay, so like the C from DNA, deaminase. So it can deaminate your cytidine. So that's up here when we said chemical changes, right? These are oxidative deamination. So it's taking that amine group and taking it off. Okay, and replacing it. And so what you can do is you can use that to make your cytidine look like uracil, okay, and it's going to make things look different. And what you can do is, we call that somatic hypermutation, okay? It can cause a ton of changes in the actual DNA structure. And this is one of the few instances where you would like that to happen. AID is very tightly controlled because you don't want it to just run around your DNA doing that. And so that's an interesting idea in and of itself, like how do you actually control it? But it can induce tons of mutations in the DNA, which is going to increase the actual diversity of amino acids in the binding sites of your antibodies, which is thus going to allow them to potentially bind even more things in the universe. So if you add up how many different genes you can grab from V, D, J, and I guess you can include C, all the antibody diversity for this binding site here, because you have two chains too, there's a heavy and a light chain, plus somatic hypermutation, I think the estimate is 10 to the 12th possible binding sites, which I don't even know. How many stars are there in the universe? It's like less than that or something crazy. It's double that. It's 10 to the 24th stars in the universe. Okay, fine. So you can, but 10 to the 12th is like an amazing number. It's like so much. That's so big. So that's really cool. I don't know. So there's another one. If you take virology with me, then you will learn. We talk about ApoBec. We've talked about that a few times this year. It's the name is super long. Apolipoprotein. Okay, so it's a protein with lipids. mRNA editing enzyme, catalytic polypeptide like. Oh my goodness. What does it do? Well, it's a cytidine deaminase, just like AID is. But what it will do, it will cause mutations to occur in like viral RNA or viral DNA in the genome of a virus. And so you keep this inside your cells as a weapon against invading viral genomes. Super duper cool. Now I'm doing like characters and stuff. Okay, we're almost done. So less nitty gritty, a little more zooming out. So there are really a couple big ways that we can talk about mutations. I'm kind of a molecular guy, so I like looking at, what's actually happening to the molecules and stuff. But you kind of need to zoom out sometimes and think about what's actually happening. Have you lost function? Okay. That could be good or bad. Have you gained function? That could be good, like, oh, I have wings now, or something like that. But that could be bad. I mean, there's always the example of there were beetles that had wings, and they lived on an island, but it got really windy, so the ones with wings would get blown off the island, but the ones that didn't have wings would stay on the island, and so the loss of function was actually a good thing, whatever. Or you could have a lethal mutation. That's generally not good because you die. The whole point being, can you go on to make babies or not, right? That's the whole goal. So that would be fitness. So function versus fitness. You can have, so these are different ideas, right? So you could have a harmful mutation, it could be beneficial, because a loss of function could be harmful or beneficial, right? So you could have a harmful mutation, you could have a beneficial mutation, it could be neutral. You could have a nearly neutral one, so that's where like I don't know, it's like nearly neutral. I don't even have a good example off the top of my head, but wait, why is this moving? I'm so sorry. There we go. So like it's like nearly neutral ones are ones that like they're not affecting the fitness, but they persist in populations. So like that CCR5 mutation example that I was giving, that would be an example of a nearly neutral one. The selective pressure doesn't really exist much, so it's not really selecting for the mutation, and yet it continues to persist. So that's kind of interesting. So that's kind of bridging the gap between like a neutral mutation and a beneficial one are the nearly neutral. So they're like potentially beneficial, I guess. Yeah. So over here on this chart, it's just some other ideas of like, how do we classify mutations? Are they spontaneous or induced? So I didn't use that term yet. I'm so sorry. Spontaneous would be ones that like your RNA polymerase or your DNA polymerase would engender. That would be a spontaneous mutation. So it's happening inside the cell from something inside the cell. An induced mutation would be something outside the cell. So DNA polymerase causes spontaneous mutations, basically. Or I suppose like if you had an intracellular peroxide or like a free radical or something, you could probably argue that that's a spontaneous one. But it's probably also induced. So induced is like, did something cause the mutation? Then it's induced. So I guess in that case, even if it's an intracellular free radical, it would still be caused by something, so it's being induced. It's not the DNA polymerase. Depends on the cell type. What is it doing to the actual function? How is it actually changing the mutation? How is it actually changing the protein? So there are lots of different ways to look at mutations, and it just kind of depends on what you're doing, or what you're trying to study. But usually we're thinking about, is it silent? Is it missense? Is it nonsense? That's typically how we're going to approach mutations. The very last concept that I would like to talk about is that not all mutations are a bad thing. I mentioned this briefly talking about like HIV. It causes a lot of mistakes because its DNA polymerase, it doesn't do a very good job. It has really high mutation rates. And so on this chart down here, so this is a 10 to the negative 8. So this is less mutations, more mutations. And so these are all different viruses up here. Okay. And so in the blue are DNA viruses. And so you tend to, if you're a DNA virus, you don't want as many mutations because it's harder to fix. You're not making it as many babies typically. And then with your red dots up here, these are your RNA viruses. And so they have RNA-based genomes, and you cause tons of mutations because you're going to make a ton of babies, you're going to have tons of selective pressure. And so let's say 1% of your offspring actually survive to make offspring of themselves. Well, that's fine if you have 10 billion offspring per cell or whatever it is. And so you can afford to make some mistakes and cause some mutations in the hopes that you would accidentally make a beneficial mutation. And you can see here, so we would be in here, higher eukaryotes. So here's how big our genome is. Okay, we have a pretty big genome. And then your mutation rate, so the less mutations on the bottom, more mutations on the top. And so you can see as we exit and we get less and less complicated, we actually see more mutations happening and the mutation rate increasing because that's basically you're trading the ability of your offspring to, the number of offspring to survive, and you're making just more offspring in the hopes that any selective pressure you will accidentally mutate against. And so that's what this is trying to show you. Pretty fun. So that's mutations. Next week, we're going to be talking about mutation repair. And so a lot of this stuff is going to come back up.